Let's get started.
Okay.
Okay, thanks everybody to come and join this seminar. It's my great pleasure to introduce Dr. Mark Wu, who is a professor and a physician at Johns Hopkins University School of Medicine. Mark is a leading neuroscientist whose work has significantly advanced our understanding of sleep regulation as well as circadian biology. Mark earned his MD-PhD degree from Baylor College of Medicine, where he studied how cinema transmission is controlled in Hugo Barron's lab using Drosophila. And then he completed his neurology residency at UCLA. And after that, then he start to study sleep by pursuing a sleep medicine fellowship at the University of Pennsylvania, where he worked with Amita Segal's lab to really study how sleep is controlled and regulated in Drosophila. And Mark became a faculty at Johns Hopkins in 2009. And then the research in Mark's lab using now both fly and mice to really study the molecular and circuit mechanism underlying sleep regulation. And in over the past decade also, the research in Mark's lab has produced numerical high impact publications and has shaped a lot of new our understanding of how sleep is regulated. In particular, the lab characterized a gene called wide awake and try and see how does that serve as a conserved molecule to link the circadian to the sleep timing. And today he's also going to talk about his new, very cool, exciting work in the lab to actually really understand how a brain structure or brain circuit re-encoding the sleep drive that many of us experience when we stop very late overnight. Okay, so without further ado, please join me welcoming Dr. Mark Gu.
All right. Thank you very much for that kind introduction, Han. And again, thank you for the invitation to speak here today. It's really a pleasure to be here. University of Wisconsin-Madison has such a rich history of genetics, particularly fly genetics, with people like Barry Ganetsky, William Engel, Sean Carroll, Jerry Yin. And of course, my lab, I kind of trained as a Drosophila geneticist and Drosophila neurogeneticist. Although today's talk actually will not be about fly genetics, but rather will be about sleep. And of course, at University of Wisconsin-Madison, you always also have a tremendous research program in sleep led by Chiara and Giulio. So today, what I'd like to tell you about is some recent work from my lab on a new sleep circuit that we argue encodes homeostatic sleep drive, and particularly how plasticity of the circuit is what really encodes homeostatic sleep drive. So most neuroscientists study what the brain does when animals are awake. But in fact, all of us and animals throughout the animal kingdom spend a lot of time sleeping, a lot of their lives. All of us are going to spend more than 20 years of our lives asleep. But what is sleep for, and how is it regulated? So, as I mentioned, we know that sleep is highly conserved throughout the animal kingdom, and so clearly plays some important biological role. It could be seen in animals as simple as worms, like the C. elegans, or even, and of course, fruit flies, which is one of our favorite model organisms, and humans, and mammals, and vertebrates, fish. And some people have argued that sleep can be seen even in organisms as simple as jellyfish that don't have a brain, but instead their nervous systems are more like a neural network. So sleep is highly conserved in the animal kingdom and must be doing something important. Another thing that really underscores the importance of sleep is the unusual evolutionary adaptations that have evolved in order to preserve sleep. So, for example, and many of you may have heard of this example, but some of the students may not have. So, for example, there's this phenomenon called unihemispheric slow wave sleep. So, marine mammals like this beluga whale or this bottlenose dolphin have a problem. They breathe air, they have lungs, but they live in the ocean. And so they have to surface to breathe every 20 to 30 minutes or so. But how do you accomplish that while also sleeping at the same time? So the way they accomplish this is they actually will put half of their brains to sleep at a time. The one half of their brain will sleep, and the other half will control their movements so that they can then surface to breathe air, and then it'll flip-flop. So this can be seen in these plots here. So these plots are plots of slow wave sleep. For example, this beluga whale, you can see the right side of their brain and the left side of their brain as measured by electroencephalography, EEG. So this is a plot of the power of the slow wave activity. And what you can see is that when this beluga whale is asleep, the left side of their brain is asleep, and then the right side of the brain is asleep, and then the left side of the brain is asleep again. And so it can flip-flop. In contrast, in rats or mammals, other mammals like us, you don't have this phenomena and you have perfectly symmetric, essentially sleep throughout the night, or in the case of rodent during the day, probably. All right, so this is just an example of unusual evolutionary adaptations that occur that really underscore how important sleep is. So let's talk a little bit before we get into the science. I just want to give a general introduction. And first, I just want to introduce stages of sleep and how we quantify or characterize sleep in mammals. So in mammals, we predominantly use something called EEG or electroencephalogram graphy, which is basically field recordings of your brain. And this is a kind of a hypnogram of humans here. So you can see what happens in humans. There's different stages of sleep. There's wake, then there's REM sleep, and we're going to talk about the different stages of sleep in a moment. But there's REM sleep and there's non-REM sleep. And then it cycles through these different stages, and you can see REM sleep occurs, every 90 minutes or so, multiple times throughout the night. So let me spend a minute here to talk about the different stages of sleep and how they look on EEG, because this will be relevant to our talk later. So broadly speaking, there's two kind of classes of sleep: rapid eye movement sleep and non-rapid eye movement sleep, or non-REM sleep. So rapid eye movement sleep is the period of sleep when you're dreaming, when you have those vivid dreams, your eyes are moving back and forth, and your muscle tone is low. Non-REM sleep is the period of sleep that can be divided into light or deep. But for today's talk, we're really just going to focus on deep non-REM sleep. In mammals, I mean, in mice, we pretty much only score essentially, deep sleep anyways. But so what does this look like on brainwave activity? So the non-REM slow wave sleep is the restorative form of sleep, and that's the part of sleep that tends to occur earlier in the night, and REM sleep tends to occur later in the night. Okay, so what do these stages of sleep look like on brainwaves or your EEG? So during wakefulness, your brainwaves have mixed fast frequencies, and that actually looks a lot like your EEG during REM sleep. So your REM sleep EEG looks a lot like your brain when it's awake. You have mixed fast frequency. It tends to be lower amplitude, and you can also see things like sawtooth theta waves that don't tend to occur during wakefulness, but it's easy to confuse these two stages. But as I mentioned, during REM sleep, you also have the eye movements and the low muscle tone. During slow wave, deep non-REM sleep, what you have are these high amplitude slow waves. These delta oscillations are 0 to 4 hertz. And you can see here, this is what we're really going to focus on today, because this is really because slow waves, as we're going to talk about in the next slide, are a measure of sleep need, sleep depth, and also sleep need. So we've known for like 50 years or more than 50 years, that slow wave activity during sleep is really a good marker for sleep need. So for example, these are some really old plots from the 1970s, and these are of humans. And so what you can see is that as you sleep, slow wave activity or slow wave sleep decreases with increasing sleep time. And conversely, it increases with the longer you are awake. So sleep need is really is thought to be reflected by slow wave activity, that the power of the high amplitude slow waves during uh, non-REM slow wave sleep. Now, just, to take a step back, just to remind the students that sleep in broad strokes is thought to be regulated by two main forces, a circadian force and a homeostatic force. And this is a very influential model, an early influential model developed by Alexander Borbelé, where he called the two-process model. And basically, during this two-process model, the propensity to sleep is thought to arise from interaction between the circadian process, process C, and the homeostatic process, process S. And the mathematical variable that he used to model process S was indeed slow wave activity. Okay. And so what you can see is these two things interact. So when process S is high and process C is not driving arousal, then you get a higher propensity to sleep. And then process S goes down as sleep occurs, and then it goes up and keeps going up if you have sleep deprivation. Okay. And also, just to remind the students, although I'm not going to be talking about it today, the circadian pacemaker is known to be housed in the suprachiasmatic nucleus in the anterior hypothalamus. It's a really beautiful structure, and it's an amazing structure because this small group of neurons, in maybe about 10,000, 20,000 neurons, really encodes time, biological time. And you can take this structure and cut it out and stick it on a dish, and it'll still keep time really for weeks. So this is an example of that just to show the students. This is a PER2, this is a clock gene promoter driving luciferase, which is a bioluminescent protein. And this is an SCN slice in a dish. And what you can see is these beautiful synchronized rhythms that occur on a 24-hour pattern in a dish that persist for weeks in the absence of any cues. There's nothing there, just it's in a dish, and it just keeps time beautifully, the clock. Okay, but anyways, we're not going to be really talking about circadian mechanisms today. What we're going to be talking about instead of homeostasis. So before I talk about sleep homeostasis, I just wanted to take a step back and introduce the concept of homeostasis more broadly. For many of us in biology and neuroscience, homeostasis is this principle that's so important and it's so kind of intuitive now to all of us, right? But in fact, it was actually not until Claude Bernard, a French physiologist, really articulated this idea, and he didn't even call it homeostasis. He argued that he kind of really pointed to this importance of stability of the internal, what he called the internal milieu, in order to deal with kind of changes in the environment, kind of a dynamically changing environment. In order for animals to survive and thrive, they needed to be able to keep their internal milieu, as he called it, stable. And Walter Cannon, who was an American physiologist then significantly later, then really coined the term homeostasis and basically argued that this is a process which is self-regulating by which biological systems maintain stability but are able to adjust to conditions that are optimal for survival. So I want to talk about this idea of homeostasis a little bit just to draw a contrast to what we're going to be talking about later, which is sleep homeostasis. A lot of homeostatic properties in the human body or in animal physiology, and things that certainly Bernard and Cannon were generally thinking about, occur without conscious effort and occur on a kind of rapid and acute time scale. So for what do I mean by that? So for example, think about oxygen. Oxygen is kept within a very narrow range in your body, right? If your oxygen goes down, your brainstem will trigger you to breathe. This occurs without you thinking, and it occurs on a very rapid time scale, very kind of quickly, right? This kind of loop is very fast and it's very transient. Okay. In contrast, there are also other kinds of homeostatic drives, which are behavioral homeostatic drives that operate on a different kind of time scale. And so here I want to highlight Kurt Richter, who many of the neuroscientists may know is a pioneer in sort of experimental psychobiology. He was well known for having spent his entire career at Johns Hopkins, almost like 60 years, and made many seminal discoveries, including pointing towards the anterior hypothalamus and the suprachiasmatic nucleus of circadian pacemaker. He studied lots of things about how there's sort of like nutrient-specific needs and really developed the use of rats as a model system. But anyways, one of the things that he studied that I want to highlight is this idea of behavioral homeostatic drives And what do I mean by that? So take, for example, hunger, right? Thirst or sleep. These are also homeostatic systems, right? If you don't eat, let's say, for 12 hours or 20 hours, you're going to feel hungry, right? And then you're going to want to do a behavior. You're going to have a motivational drive to satiate that behavior by eating a pizza, for example, here. Same thing with sleep. You also accumulate the need to sleep. All of us have had the experiences if you stay up all night, right? Then you feel sleepy the next day, right? So this is kind of a homeostatic drive, and then there's motivation to then go to sleep. So the things I want to point out are that for these behavioral homeostatic drives, they operate in a different time scale than those other metabolic and cellular drives that I told you about, or homeostasis I told you about on the prior slide. They occur more gradually, and the need or drive accumulates gradually, and the replenishment or repletion of these drives have to occur over a time frame. In other words, you don't just take one bite and then you're done, right? You have to satisfy the drive and you have to do it for some time. Same thing with sleep. You don't have a system that our system is not designed where you feel sleepy after 1 second or a minute, and then you sleep for 1 minute and then you're good to go. That's not very efficient. As an aside, just to, point out a random thing is that when I was doing this, finishing this talk, I asked AI to make these pictures for me. So I asked it to give me a picture of a person eating a pizza, and this is kind of what it imagined, this person wearing a casual shirt with a soda and eating a pizza. And I asked it to, show a person sleeping. But one thing you can realize this was AI generated is because there's always these slight errors, right? So like if you look at his t-shirt, it doesn't make any sense. Like it says Niey, like I don't even know what that means. I don't think there's any bands named Naey something. And if you look at the clock, it says 9:44 p.m., which is when I was doing this talk, but then it's like sunny outside. Anyway, so I just want to point that out for people that are using AI for their own purposes, that you always have to watch and make sure it's not making funny errors. So anyways, back to science. So um, one of the questions, in neuroscience really is how are these behavioral homeostatic drives, these motivational drives generated and persist over time? So now that leads us to sleep homeostasis. So as I mentioned in the previous slide, sleep is a classically homeostatically regulated behavior, but how sleep is homeostatically regulated is not very well understood. One of the most early influential models was the somnogen hypothesis. And this was in the early 20th century, in the early 1900s. Henri Péron and this another scientist in Japan, Ishimori, did these experiments where they basically took animals, dogs, and I think also goats and pigs, and what they would do is sleep deprive them, right? Keep them awake. Then they would withdraw cerebrospinal fluid from these animals and inject them into control animals, like you would inject them into another dog. And what happens is those dogs then fell asleep. And so they concluded that the way sleep homeostasis worked was there were these circulating substances that accumulated with sleep need and then triggered sleep. And this is called the somnogen hypothesis. And over the past 100 years, a large number of substances have been implicated in this process, the best known of which is adenosine, but also things like prostaglandins, various immune cytokines, and others as well. More recently, keeping in terms of staying with a molecular focus, a number of scientists have argued that phosphorylation of synaptic proteins or kinase activity in its synapses is important for the homeostatic regulation of sleep. Um, so, people like Chiara and Giulio have shown over the years that, in fact, synaptic proteins can be phosphorylated with sleep deprivation. But what started this kind of model or this proposal was Masashi Yanagisawa did this large screen of mouse mutants, about 8,000 mutants, and found a mutant that he called Sleepy. And this Sleepy mutant in mice with a single copy, so it's a dominant mutation, right? Which, basically the phenotype was when you look at EEG, is that they had high sleep. And remember, these are mice, right? So mice tend to normally sleep during the day and they tend to normally be awake during the night. The sleepy mutants had high sleep all the time, both during the inactive period, but also during the active period. And this is true whether you're a heterozygote mutant or a homozygote mutant. Interestingly, these mutants also showed high delta power or slow wave activity. And remember, we had talked about how delta power and slow wave activity is a measure of sleep need, right? And so these mice, despite the fact that they sleep a lot, also had high delta power, which, as an aside, as a sleep clinician, I will also mention that as a sort of a human parallel, sometimes when we see patients with a disease called idiopathic hypersomnia, they also have really high sleep time. And interestingly, when they sleep, they still have really high delta power. So it's kind of like echoes this kind of human condition. So it turns out, anyways, that this sleepy mutant is a gain of function mutation in a kinase called SYK3, which is salt inducible kinase 3, which is in the AMPA kinase family. Then a substantial body of work, done by Qinghua Liu, Masashi Yanagisawa, and others, have led to this model that there's a lot of proteins that are phosphorylated at the synapse with sleep deprivation, and in many of them may be direct targets of SYK3. And this phosphorylation and this activity, this phosphoproteome at the synapse, really might be encoding sleep drive. In addition, Hiroki Ueda has, as we'll talk about a little later, has also argued that certain kinases play a key role in the homeostatic regulation of sleep. Okay, so what my lab has been doing is taking a different approach, which is asking the question of whether circuits or neural circuits may be important for sleep homeostasis. So that's people have been really focused on the molecular aspect, and we've been thinking about it from the network or a neural circuit perspective. So what I want to just point out is that, or this just introduce think about is sort of you can think about homeostatic systems in engineering terms as a negative feedback loop with a sensor, an integrator, and effector. Okay, so a good example of this kind of a system might be your central air conditioning or heating unit. It's probably pretty cold there in Madison. It's certainly quite cold here in Baltimore, so you probably have your heater on, right? So how does that system basically work? So hey, there's a temperature probe somewhere in your system. It says, hey, the temperature is too low. This information is sent to an integrator circuit that takes that information, compares it to a set point, and then directs an effector to change the temperature, turn on the heating element, for example. Then by doing so, the temperature starts to rise. This information is sensed by the sensing and so on and so forth, right? So it's very intuitive to understand this kind of process. So one could imagine that sleep might also have such a kind of a system, right? That somewhere in the brain, sleep need might be sensed. What what cellular components might be sensing sleep need is certainly not known and open to debate. We and others, including Marcos Frank and other people, have suggested that maybe astrocytes might act as sensors for sleep activity. Somehow, this sleep information or the sleep state variable has to be sent to some circuit that might have to tell other circuits what to do. We would argue that many, as we'll discuss in the next slide, many of the sleep circuits that have been identified to date may fall into the role of what we call effector circuits or switches, basically. And I'll explain a little why we think that. And so one of the things that we sought to identify was circuits that might act upstream of these effector circuits and might have a role in somehow computing or encoding sleep drive. So one of the key questions here, which I'm just going to leave as an open thing for now, is that one of the key questions, right, is what is the state variable for sleep? What is the thing that's being detected to encode sleep need? In the case of this, central AC unit that I was talking about, it's temperature. It's very easy, right? But what is it that is encoding? What is the state variable sleep need? Okay, anyways. As I was alluding to, there are quite a few non-REM promoting circuits that have been identified over the years. I think the count now we've done is somewhere close to 20 or 30, something like that, non-REM promoting circuits, depending on how you define them. So there's a lot of them. The vast majority of them are inhibitory, although there are a few that are excitatory. And like I said, these all tend to have the same similar phenotype, which is the following. And this is also why we think that these kind of function as effector circuits. Basically, if you take all of these previously identified circuits and you activate them optogenetically or acutely, the mice will quickly go to sleep. And then if you stop activating if you turn off your optogenetic stimulation, then they wake up. So in that sense, these circuits kind of function like a switch. Um, so anyways, and then that'll lead us into, kind of what we did. But the other question you may be asking is, why are there so many sleep circuits, right? Because I told you that there's only one suprachiasmatic nucleus, which is the master pacemaker. Now, granted, there are, to add some complexity to that concept, there are local brain clocks and local clocks with the body, but there's certainly, everyone would agree that the master pacemaker is in the SEM. So why are there so many sleep circuits? And this is an open question in the field, but one of the things that I think and others think maybe is that these sleep circuits may listen to different inputs. For example, some circuits, some sleep, and that might reflect varying and multiple functions of sleep. So, for example, there are sleep circuits that respond, for example, specifically to stress. They are activated by stress. They are, then they sort of listen to stress, are activated, and then they kind of promote sleep following stress. That's kind of an example. Anyways, okay. So now on to our work. So this work was led by a talented postdoc, Sang-Soo Lee, who's currently in the job market. And what he wanted to identify an upstream circuit that might kind of encode sleep drive. It might coordinate the role of multiple downstream circuits and would serve to excite them. So based on this concept, he embarked on a very risky and labor-intensive work project. So what he did, and this is a bit of a complicated panel, I know, but what he did was take 11 known non-REM sleep promoting circuits, okay? And he did this in a cell type specific way. What I mean by that is, for example, in the zona incerta, he specifically labeled LH6 positive cells by using LH6 Cree. Okay, so all of these 11 non-REM promoting circuits are done in a cell type specific reason, not just region specific. Then what he did is he did rabies virus retrograde labeling to label brain regions that were upstream of those 11 regions. And he identified about 140 brain regions in the mouse brain that are upstream of those regions. From those 140 regions, he then narrowed it down to about 11 or so, shown here. And I'm not going to go through what each thing means, but like this is like lateral habenula, and of course, this is the reunions nucleus we'll be talking about. So you narrowed it down to 11 regions that had very dense kind of like labeling, retrograde rabies virus labeling, and that seem to project to multiple sleep promoting circuits. And that's what this plot really reflects, that these brain regions have projections to multiple sleep promoting circuits. And he kind of subjectively quantified them from like sparse to dense in terms of a heat map. Okay. So then he basically anatomically came up with these 11 regions where there's excitatory neurons that are upstream of sleep promoting circuits. Okay. And then what he did was a functional screen where he did chemogenetic activation. So I think you guys are familiar with chemogenetic activation, where you use DREAD, and then you inject CNO, and then you can chemogenically activate the neurons. So he then did chemogenetic activation for all 11 of these circuits. And this is shown here. And what you can see, he used galanin expressing VILPO, ventrolateral preoptic nuclei neurons, as a positive control. This is a well-established non-REM promoting circuit. And the circuit that had the largest non-REM increase was in the thalamic reunions nucleus, which had never been previously implicated in sleep. So he thought, okay, that sounds interesting. So he started to pursue this in more detail. One of the things that was really interesting about this RE circuit was not only that it increased non-REM sleep by a lot, but the time course was very unusual. So what do I mean by that? If you look at this bottom left panel, as a control, we're talking about galanin positive VILPO neurons, right? So if you chemogenetically activate them, of course, chemogenetic activation takes like 30 minutes to an hour for the C node to get into the body, and then, kind of then takes effect over the next few hours. But the sense you get, what you can see is that VILPO activation, chemogenetic activation, leads to an increase in non-REM sleep quite early, and then it rapidly tapers off. In contrast, chemogenetic activation of RE neurons did not increase sleep for literally hours. It took like several hours before it started to go up. And then it lasted for a long time, like almost like, look at this. It's like 6, 8 hours. The injection was here at ZT12. ZT stands for Zeitgeber time, which is time after light's onset. So 12, here's the injection, and you've got sleep persisting with the activation of chemo activation of RE for, 12 hours, it seems like. So that was very unusual. And another thing that was unusual was that when you look at the delta power, remember that delta power is a sense of sleep need of this chemogenic activation of RE. Interestingly, the time course of the sleep need activation occurs much earlier. So at 2 hours after the CNO injection, the delta power has already gone up. And as the sleep goes on, then the delta power comes down. So the delta power increase precedes the actual sleep. In other words, these data suggest the idea that chemogenetic activation of RE neurons might induce sleepiness rather than directly switching the animal into sleep. Now, having said that, we all know that chemogenetics activation is really bad for temporal resolution, right? So, oh, actually, let me take a step back and just explain a little bit about the RE. So some of you may not be familiar with the RE. I certainly wasn't before we started this project. So the RE is a specific nucleus in the thalamus. It's called the nucleus reunions. And some of you may have heard of the nucleus reunions in terms of its function regulating, spatial navigation and memory through projections with the prefrontal cortex and the hippocampus. Those circuits or those parts of the RE are in the anterior aspect of the RE. Whereas what we're talking about, the sleep regulating RE neurons are in the middle of the RE, and no one has studied this region as far as we can tell. And certainly no one has implicated this region in sleep regulation. Because no one has really studied, we kind of made up the names mid RE and anterior RE, but I think we're the first people to call it that. I just as an aside, this reunions in the mouse brain, it kind of looks like the Starship Enterprise in Star Trek. It has this kind of flat surface in the front, and it's kind of got this tail in the back. Okay, anyways, and as I mentioned before, these RE neurons in the mid region have projections to many sleep promoting circuits, most prominently to the zona incerta, preoptic area, ventrolateral preoptic nucleus, and basal forebrain. Okay. So anyways, getting back to what I was saying before, which is that we all know that chemogenic activation has poor temporal resolution. And so we next did optogenic activation. You all are familiar with this, where you use blue light, you shine blue light, and there's a channel rhodopsin, and you kind of, can force the neuron to fire, right? So this is what happens. So when you give a 5-minute pulse of blue light firing at about 20 hertz, I think this was a 20 hertz experiment. Interestingly, nothing happens to sleep wake for 20 minutes. So this is really different from all the other sleep discs I told you about, right? Because when other people activate like if you activate Vilpo, they just go straight to sleep. They'll be sleeping during the 5-minute activation, but nothing changes with sleep for 20 minutes. Then, long after the optostimulation is over, then they decide to sleep for about an hour. This sleep is deeper because it has increased delta power shown here. It's also plotted here, but anyways, you can just draw your attention to here. So this sleep is more consolidated. I didn't show you the data because it makes the slide kind of clunky, but it's more consolidated and it's deeper. So this kind of deep, consolidated, persistent sleep reminded us of the kind of homeostatic recovery sleep that occurs after sleep deprivation, right? If you stay up for a long time, you're going to get deep, consolidated, persistent sleep. So the next question is, what is going on? So I asked, Sang-Soo, what do you think is going on during these 20 minutes anyways, right? Because the first thing is, I was like, Sang-Soo, reviewers going to ask you, are these animals like super hyper or something? And that's why they're having, sleep rebound later? Clearly, they're not actually more awake because then the non-REM sleep would have gone down, right? But what are they actually doing? So it turns out that the mice are getting ready for bed. So it turns out that there's things called pre-sleep behaviors that are seen in many animal species, not all, but many animal species, particularly in small mammals, small rodents and small mammals, and some primates. So you know how when you go to bed and when you go to sleep, you have a routine. You brush your teeth, you arrange your blanket, you arrange your pillow. and then you go to sleep, right? So mice actually do something similar. So when mice go to bed, before they go to bed, they actually spend time grooming, cleaning themselves. They will do nesting behavior. They organize their nest. Um, and so this is what happens when we optogenically stimulate RE during the stimulation and for those first 20 minutes. What the mice do is it's a little bit hard to see from this distance, but basically what they're doing is kind of grooming their whiskers and cleaning. And then what you can also see them doing is organizing their bedding behavior, the nesting. So they kind of take nesting and they organize it into a bed. And this is kind of funny. We saw this a few times, but it hasn't really been described in mice. Wait, how do I? Oh yeah. So if you watch closely, the mouse kind of kicks out the poop. So it's almost like it's trying to clean up its bed before it goes to sleep. It's not a behavior that's been ever described that we've known. So so basically what's happening is the mice are getting ready for bed. They're engaging in pre-sleep behaviors or also known as sleep preparatory behaviors. It's just quantified here. So controls are in white and the experimental manipulations in color. And so what you can see is that initially there's not a huge change in locomotion for the first 10 minutes, and then then locomotion goes way down. But and then also what you can see is that while the controls are eating, they don't really eat. But what they do is spend a lot of time grooming. So basically, once we activate the RA neurons, they decide to start grooming. So they're not like running around like crazy or anything, but they're just grooming and also nesting, right? Doing nest building and nest organizing behavior. And then over time, after 10 to 20 minutes, then they stop moving. And immobility here was scored as when the mice are not moving, but also in a stereotypical sleep posture, which for mice is basically like their tail is curled around their body and their head is like tucked down. Their nose is kind of tucked down. This is also can be shown in this kind of a way, in a sort of a top-down spatial way. So um, what happened? This is just to show you where the mice are, when the RE is activated. So the yellow is a nest, okay? And this is a control. This is an experimental animal. Once we start activating the RE, the mice head straight for the nest, essentially. I mean, they walk around the cage once, essentially, and then they go in the nest. And basically what you can see is they spend the entire rest of the 20 minutes while they are not asleep, right? We talked about how they don't go to sleep until 20 minutes later. They're not asleep, but they're just sitting in their nest. They're grooming, they're nesting. Controls never sit in the nest, right? They're like, you know, walking around and like doing some stuff and then walking around, but they're never in the nest. And that's also quantified here. Okay. So the next question we wanted to ask was, what what do these RE neurons do? How do these neurons fire, or what is their activity pattern during sleep deprivation or changing sleep need? So we collaborated with, so many of you are familiar with NeuroPixels, but for those of you that aren't, NeuroPixels are basically like this it's a new invention where it's a high density silicon probe that allows for simultaneous recording of thousands of neurons simultaneously, where you can measure their spiking activity, and there's like four shanks that go down. NeuroPixels 2.0 is just a lighter version of NeuroPixels 1. This technique was developed by Tim Harris. And so we collaborated with Tim and people in his lab, in his group, to do NeuroPixels recordings of MRA activity during sleep deprivation and 6 hours of sleep deprivation and 6 hours of recovery sleep. So what we have are chronic NeuroPixel recording in freely moving mice. We're also recording video, and they have a nest. Okay, and then post hoc we make sure that the shanks were in the RE. So because the data sets are so humongous, we only recorded 10 minutes from each hour. Okay. And so there's kind of like 12 recording sessions, each 10 minutes long, once an hour, right? So 6 recording sessions during SD, 6 recording sessions during recovery sleep. What you can basically appreciate here in this plot to the bottom left is on average, RE activity is elevated during sleep deprivation and reduced during recovery sleep. These RE neurons are firing on average about 15 hertz during sleep deprivation and about 5 hertz during recovery sleep, quantified here. Also, just to give you a more granular view, this is a heat map plot of these data. We've recorded about, I think we got somewhere about 150 or 200 neurons. So each row is a different neuron. Each bin is a different session, and the color reflects how fast they're firing. Okay? So what you can appreciate is that maybe that a substantial portion of the network population, maybe about half, have really fast firing during SD or sleep deprivation, and then are quiet during recovery sleep. In contrast, there is a population, maybe about half, that doesn't seem to change. Okay, so the next question we asked was, okay, so what we learned from this is that these neurons, unlike most non-REM promoting circuits defined to date, are activated with wakefulness and SD, not sleep. Second thing we learned is there's two populations, right? The third thing we also learned, this is a finer point, but those in the sleep field would appreciate. The firing rate, contrary to our expectation, did not go up with increasing sleep need, right? The idea is if the RE neurons are directly encoding sleep drive, then their activity should go up with increasing sleep need, but it doesn't. It stays flat. And so we'll get back to that later. So the next question was, are these wake active RE neurons important for promoting sleep or promoting recovery sleep? And so we used the FOSS-trap system, some of you may be familiar with. This was developed by Leach and Low. Basically, what it is the FOSS, which is an immediate early gene promoter tied, which drives expression of a Cre-inducible Cre-ERT2. This is a Cre recombinase that is only active when you also provide tamoxifen. And then when you provide the tamoxifen, which we did during sleep deprivation or prior to sleep deprivation, then you can excise things and you can label neurons, but you can also, by injecting Cre-dependent viruses, also induce expression of various effector proteins like DREG-GQ for chemogenic activation or caspase for ablation. In brief, what I want to say is that indeed, it's the wake active MRE neurons that are critical for this homeostatic sleep process because if you chemogenically activate just these SD trapped cells, you can increase sleep. If you ablate these SD trapped cells, you can significantly reduce homeostatic recovery sleep following 6 hour sleep deprivation, and you also get a reduction in delta power. So it is the population that are wake active that seem to be important. So next we wanted to ask, okay, so now we know that the RE neurons show higher activity, right? They triple their firing rate during SD, but is that activity important for subsequent homeostatic rebound? So the way we did this experiment is we did chemogenetic inactivation or inhibition of the RE neurons, but we did this in a way that's, I think, different from what other people had previously done. We chemogenetically inhibited these neurons during sleep deprivation. So we injected CNO at the onset of sleep deprivation, right? Because this is when the neurons are firing faster, right? We did it here, and then we looked to see what happened to subsequent homeostatic recovery sleep. So when we reduced firing during sleep deprivation, you got a reduction in the amount of homeostatic recovery sleep, as well as the delta power, again, which is a measure of sleep depth and sleep need. In contrast, if you do the same protocol to Galen-Vilpo cells, which is a well-known non-REM sleep promoting circuit, you get no effect. So this is very interesting because it suggests that this activity, this increased activity of the RA neurons is important for accumulation of sleep need or drive. I should say when other people, I think this may be the first time this has been done in the field, but when people generally do this, people generally do the inhibition during the recovery sleep and then argue that these neurons are important for homeostatic recovery sleep. But what we've done is do it actually before the actual homeostatic recovery sleep. Okay. Next question is, where do these RE neurons act, right? So we proposed that they should act on a non-REM sleep promoting circuit. That was indeed how Sang-Soo designed his screen. And so that's the next question we tackled. So Sang-Soo decided to focus on two places, the zona incerta, because there were particularly strong projections there, and also ventrolateral preoptic area. So what he did that next did is did rabies virus mediated channel rhodopsin. So he injected our rabies virus channel rhodopsin into the downstream circuit, either in the zona incerta or in the ventrolateral preoptic nucleus, which is galanin positive. Again, he did this in a cell type specific way. Then he shined the blue light on the RE, so he's activating this population specifically. If you activate the RA neurons that project to the ventrolateral preoptic nucleus, you basically get no effect whatsoever on sleep. If you optogenically activate the RA that project to the ZI, the zona incerta, you get exactly what we showed in the beginning, which is a 20-minute delay, and then you get an hour of persistent sleep that is deeper. These data suggest that the RA neurons act on the LH6 positive ZI cells to promote sleep. And this was very convenient for us because Seth Blackshaw had published that the LH6 positive ZI cells were important for promoting sleep, and he's my frequent colleague and neighbor, so we were able to collaborate with him to study that further. The next thing Sang-Soo did was to look at, he did epistasis experiments. So of course in genetics, you guys think a lot about genetic epistasis. You can also do circuit epistasis, and that's kind of what Sang-Soo did in this set of experiments to prove that it really was the LH6 positive ZI cells that the RE neurons acted on. So what he did was, again, he optogenetically activated the RA neurons by expressing CAMK2 alpha channel rhodopsin in the RA neurons, while simultaneously ablating the LH6 positive ZI cells. And the way he did that was inject either a control virus or a Cre-dependent caspase-3 virus into LH6 Cre mice in the ZI. Under control conditions, basically do control, basically you get this, again, this nice 20-minute delay, which they're presumably doing pre-sleep behaviors with a persistent deep sleep. And then, however, if you ablate or largely ablate the ZI, you can markedly attenuate this persistent and increased sleep, as well as the delta power. These data strongly suggest that the MRA neurons act on the LH6 positive ZI neurons to promote sleep. So remember how I said earlier that one of the things that we were a little puzzled by was what in the RA neurons are encoding the sleep drive, because sleep drive should be scalable, right? We know that if you are sleep deprived for longer, that you should then feel more sleep drive. You should have more sleep rebound, right? But it's not being encoded by the neural activity itself, because as I showed you, no matter if you are sleep deprived, if the mouse was sleep deprived for 1 hour or 5 hours or 6 hours, the firing was always 15 hertz. So what could account for the scalability of the sleep need, and what could be directly encoding the sleep drive? So one potential mechanism was something that turned out that when Sang-Soo was messing around with these optogenic experiments, he realized, because Chadwick Dobson has GFP, and when he was doing this SD and then looking at the sections, he realized that there was the RE neurons, the projections, there were a lot more of them with sleep deprivation. And it was quite striking. Um, Although I should also add that this is a very hard thing to study. It's very labor intensive and very annoying. So kudos to Sang-Soo for powering through all of this. So this in this experiment, what he does is he expresses AAV Cre-dependent MGFP, which is going to label, excuse me, label the processes with 2A synaptophysin mRuby. So this is going to label synaptophysin, which you guys may know is a synaptic vesicle protein, in red. Okay, so basically the green is going to label kind of the processes, and the red's going to label kind of like the synapses, essentially. And what he found was that that with sleep deprivation, you get a marked increase in synaptophysin particle number, which is reduced following recovery sleep. No change, though, with the circadian time control. So in other words, from ZT6 to ZT12, that is 6 hours after lights on or 12 hours after lights on, there's no change. But it goes way up with sleep deprivation, and then it goes down with recovery sleep. Similarly, he quantified the processes, the green signal, and that also markedly increased with sleep deprivation, and then significantly came back down with recovery sleep. So this was interesting because it suggested that these terminals, these RE neurons, show plasticity with sleep deprivation, with increased sleep need. And it's interesting because, to our knowledge, people hadn't really shown in mice that sleep circuits undergo plastic changes with sleep deprivation. So is this plasticity encoding sleep drive? Well, the first way he wanted to address this question was ask a correlation question, which is, if it encodes sleep drive, then in principle, the amount of plasticity should correlate with the amount of rebound, right? And so what he did was do this experiment. He repeated that experiment. Remember the experiment where he did chemogenic activation during sleep deprivation, where he injects the CNO at ZT0 at the beginning of sleep deprivation. Then he does EEG recordings. Then he does to analyze the rebound sleep. And then he does samples to quantify the plasticity. What he found was that, as he had previously shown, that if you chemogenically inhibit RE neurons, that you get a reduction in subsequent homeostatic recovery sleep. But you interestingly also get a reduction in plasticity here. And in both vehicle and CNO conditions, CNO more prominently because with CNO you get more of a range, more of a spread. There's a good correlation between the amount of plasticity as measured by synaptophysin particle numbers and the amount of recovery sleep. Okay. So that's nice, although of course that's just correlation, and later we'll try to get to some functional manipulations. The next question is, we noticed that there was a lot of plasticity, particularly around the ZI. So then Sang-Soo asked, does this plasticity change the connectivity with the ZI? And one of the reasons we asked this, because we knew at baseline, we knew from some of our experiments that the baseline connectivity between the RE neurons and the downstream ZI neurons is really poor and weak. So the first thing he did was use GRASP. So some of you are familiar with this. It was developed in worm, used in flies, and now more recently in mice. GRASP stands for GFP reconstitution across synaptic partners. And basically what you do is you express half of GFP on the presynaptic side, half of GFP on the postsynaptic side. And when you have a synapse where it's really close together, then you can see green signal. Okay? And so that's what he did in these mice, expressing half in the RE neurons and half in the ZI neurons. And what you can basically appreciate is with sleep deprivation, there was a significant increase in GFP positive signals shown here and then kind of blown up here. There were not only more of them, but they were also bigger. So suggesting that this SD or deprivation dependent plasticity increased physical connectivity between the RE neurons and the LH6 positive ZI cells. The next thing he wanted to ask was, does it increase the functional connectivity? And so the answer is yes. So the way he did it, we did two different ways. The first way we did it was optogenetically activate the RE neurons and measure response in the ZI cells by measuring FOS positivity. So 30 minutes, I think like 30 minutes or an hour later. So, and then he did in two conditions, no sleep deprivation or sleep deprivation. And what happens is when you basically do sleep deprivation, you get a lot more FOS positive responsiveness from the ZI compared to controls. The other way we did this was just with ePhys, opto-ePhys, where we did this in slices, and we optogenically activated the RE neurons, and then we recorded from ZI cells. And, Chang, who the postdoc who did these recordings, also could tell actually in a way whether the SD was done. So in that sense, it wasn't perfectly controlled because there's just a lot fewer like projections. But anyways, when we did these, he tried to be as unbiased as possible. And when he did these recordings, what he found was that the functional connectivity was quite poor between RE and ZI without sleep deprivation, but it markedly increased about threefold with sleep deprivation. So basically what these data suggest is that there's plasticity and this connectivity is increased between the RE and ZI with sleep deprivation. So now in the last couple slides, I just want to quickly talk about a molecular mechanism. And so, the reason we wanted to do this was because first we want to understand molecular mechanism, but second of all, we want to understand whether whether the plasticity was functionally relevant, right? I showed you correlation data, but it'd be nice to manipulate the plasticity and then show an effect on homeostatic recovery sleep. So we decided to focus on CAMK2 because Hiroki Ueda has implicated CAMK2 as being important for homeostatic recovery sleep. Plus, CAMK2 is really important for neuroplasticity. And people have shown that Hiroki showed that CAMK2 knockout reduces non-REM sleep. And other people have shown that activity-dependent autophosphorylation correlates with sleep need. So CAMK2 is this kind of 6-hexamer, and it's autophosphorylated, which then becomes active. And so first we showed that CAMK2 is in fact activated in MRE neurons following sleep deprivation by staining for phospho T286 CAMK2 in the RA with sleep deprivation, so it's activated. Then what we did is we inhibited CAMK2 by using a naturally occurring CAMK2 inhibitor called CAMKIN. And this was developed by or worked on by one of our former colleagues, Mark Anderson. He gave us the plasmid, and we engineered this into viruses. And so what we did now in this experiment, what Sang-Soo did was inject uh, channelrhodopsin. Well, channelrhodopsin GFP. Mainly, he did that just to label the terminals, and then injected control virus or the CAM-KIN, and then did SD. And what you can see is, yes, indeed, the CAMK2 inhibition suppresses the plasticity seen with sleep deprivation. If you compare this panel to this panel, maybe a little hard to see on your screen. The red doesn't really pop very well. But anyways, the plasticity is inhibited by inhibiting CAMK2 in RA neurons following sleep deprivation. And furthermore, remember the experiment we did with the functional connectivity where we optogenically activated in a I should say, I forgot to say, in a sub-threshold manner, and then and then labeled FOS in the ZI cells. Um, if you do this and you also inhibit CAMK2, you significantly reduce the connectivity to the downstream ZI cells. So inhibiting CAMK2 and RE reduces plasticity and connectivity of the MRI ZI circuit. Interestingly, if you inhibit CAMK2 in the MRA, it has no effect at all on baseline sleep. Because you might worry that, hey, you're inhibiting this really important kinase. Maybe you made the cell sick and they do all kinds of weird stuff and whatever. But there was just no effect on baseline sleep. But interestingly, instead, what happens is there's a significant reduction in homeostatic recovery sleep, both amount and delta power. So basically, our argument is that by inhibiting CAMK2, we're suppressing the plasticity and then suppressing recovery sleep. So let me summarize what I've told you today. From a circuit screen, we identified an excitatory non-REM sleep promoting circuit in the thalamic Renin's nucleus. The phenotype seen with this circuit has not been is really unusual because instead of directly inducing sleep, it leads to pre-sleep behaviors followed by persistent deep sleep. Unlike most non-REM sleep promoting circuits, MRI neurons increase firing during wakefulness. As I mentioned, most non-REM promoting sleep circuits increase their firing during sleep. We show that the activity of these RA neurons is required for homeostatic rebound. We show that they act on LHX positive ZI to promote sleep. We show that sleep deprivation induces plasticity at the MRI ZI connection and enhances connectivity, and that CAMK2 is required in these neurons for the plasticity of the MRI ZI circuit and subsequent homeostatic sleep rebound. To summarize this in a model, what we can say is that this is our model. Under baseline conditions, there is very poor connectivity between the RE and the ZI. With sleep deprivation, there's plasticity that increases the connectivity to the ZI, which then allows for sleep promoting signals to then allow for sleep. Because as we mentioned, the ZI neurons, these LH6 positive ZI neurons promote sleep and promote persistent deep sleep. The plasticity mechanism is not only just kind of a cool mechanism, but also is a way that could potentially account for the gradual accumulation and persistence of sleep drive. Because remember how we talked about how on the Richter slide, behavioral drives tend to accumulate over time and tend to persist over time. And this plasticity mechanism could be a very elegant way to solve that problem. I should also say there are not that many examples in general. If you zoom out beyond sleep, there are not that many examples of need-dependent plasticity that regulate the process. And so that's kind of fun as well. All right, and so I just want to thank the people that work. I've highlighted Sang-Soo, who really did the lion's share of the work. More than 90% of the work was conducted by Sang-Soo, a talented postdoc in the lab, who again is currently on the job market. And then there were a number of other people that contributed to this as well. And Seth Blackshaw's lab did single-cell sequencing for us, and the Harris lab did this NeuroPixels. Okay, I will stop there, and I think we might be able to do a couple questions through chat box for Han.
Yeah, thanks. Thanks, Mark, for a wonderful talk. So, because the attendants can't really speak over the Zoom meeting, so as I wrote down in the chat, if you have questions, you can directly click the Q&A section button in the bottom of Zoom control panel and type in that, and I will help to repeat the question to Mark. I guess while we're waiting, so maybe a question I have would be, so it seems like that only a small fraction of the RE neurons actually awake have been activate during the sleep deprivation. Are the one who specifically projected to just the ZI region, or those also project in other area? And does those neurons always or like a high correlation with the KMK2 positive cells? Or so how does that plasticity only occur just in this particular projections?
Right. So let me see if I might have that slide. Trying to remember if I put it here. Nope. Okay. Sorry about that. I'll put it back up. So to answer your question, um, I don't I don't think it's a small proportion necessarily that are activated by wake. NeuroPixel's data suggests that it was roughly half. Although I would say that when we do the FOSS trap with Leach and Lowe's technique, it is definitely less than half. It's maybe 10 or 20%, something like that. But that might just reflect, technical issues, limitations, right? That you can't capture every single cell that happened to be active then. So that's the first thing I would say. The second question is that you're asking, do those cells preferentially project to the ZI? And the answer is a little hard to say because we didn't do single cell labeling. So the really way to do this is to single cell label and then kind of quantify everything, right? It's a huge amount of work and technically challenging.
Yeah, yeah.
So what I can tell you is that Sang Soo, for example, did um, and that was the slide I was trying to find, which I don't have here on me, is that he quantified labeling to the ZI and to Vilpo under baseline and SD conditions. And what he sees is there's a lot of projections that go to the ZI with SD, and there's a little bit more with Vilpo, but not as much. So
Cool.
that's all I can really say. So there seems to be a bias that direction, but it's hard to answer very rigorously.
Cool. Great. There's a couple questions from the audience. So the first one from Kiara, which is first of all, wonderful talk, but at the same time asking, do you think what's the integrator system during basal sleep? So this RE is more like a sleep device integrator. So what's the integrator for the baseline sleep that build
Yeah.
the sleep drive?
Right. So that's a great question. And so, as Kiara rightly points out, this RE circuit is really specific for sleep deprivation conditions. And I should point out that it's not the only such circuit because if we completely ablate the RE neurons, you can still get homeostatic sleep rebound. It's just reduced. So there's other circuits involved as well, we think. But to specifically answer Kiara's question, first, we don't know the answer. So, I think this is an open question and we don't know. But if I had to guess, I think it might be somewhere with astrocytes and the neocortex. So that would be my guess, but I really don't know for sure.
Right. Another question from Lucas. How do you think the increase in the RE to CI connectivity cause an increase in the delta power? Is it-
Right. So that's a great question. And I don't know the answer to that. Except to say that I think that so there's a couple possibilities. A lot of people haven't very carefully, because it's a little bit annoying. A lot of people haven't carefully measured delta power when they do their sleep manipulation. Some people do, but a lot of people don't. And I can't rem I think that Seth in his Nature paper did not look at closely at delta power. So one idea would be through the activation of the ZI neurons themselves. Another possibility would be through other kind of recurrent circuits going back upstream, for example. So it turns out that there is actually, and this is part of Sang Soo's job proposal, that there's connections not only between the RE and the ZI, but the ZI also feeds back to the RE, and there's also connections between the RE and the colostrum. And the colostrum is this very interesting part of the brain that many of you may have heard about. Crick famously postulated that this was a seat of consciousness, but the colostrum is interesting to sleep people because it has been shown to generate slow wave sleep and slow wave activity. And and also integrate signals from large parts of the neocortex. Now, in the case, what Sang-Soo has specifically found is that the colloquium is upstream of the RE, but I wouldn't be surprised if there weren't might be some recurrent circuits as well. We haven't looked closely. But anyways, the short answer is we don't know. I don't know for sure, but one idea would be through the ZI, or the second would be through other circuit mechanisms, something like a colostrum.
Uh, cool. And then I guess maybe other questions. Maybe I'll ask maybe one more question about so the sleep preparatory behavior is very cool after the it's more like immediate effect of the
Hmm.
activation of those neurons. So
Right.
the does those also depend on the ZI or actually is actually complete different
Oh,
circuit
yeah.
for that?
Yeah. So that's a great question. And the answer is we believe it is not through the ZI, although we have not rigorously addressed this question. We suspect it's through the POA, but the reason why we don't think it's through the ZI is the following. I didn't show you these data, but we also did optogenic activation where you optogenically activate the RA neurons, but put the blue light at the terminal. So you only activate the um, the REZI terminal, right? And if you do that, then you don't have a delay. The animals go right to sleep. And it's still persistent, but you don't have that 20-minute delay where they're doing pre-sleep behaviors. So we believe, although we have not carefully analyzed this, and that's part of what my postdoc saying proposed to study, is that it's through a different projection. And our speculation is that projection under baseline conditions is stronger at baseline. So it's biased that way initially until the plasticity occurs, and then it goes for the ZI circuit.
Cool, thanks. Yeah, I think due to the time, maybe we'll stop here. Thank again, Mark, for this fascinating talk.